Hey guys, so let's continue with our inheritance lecture that we started on Tuesday. So last on Tuesday we learned about this guy, Gregor Mendel, and this is a repeat slide, so I'm not going to go through it all, but I just want to point out that he proposed a few interesting things. He proposed that traits, so external visible traits, can be inherited. These traits can be recessive or dominant, depending on which one comes through. And on Tuesday, we actually got to cover recessive and dominant genes. Another thing to point out here is that the visible trait, the visible part of a trait, is called the phenotype. That's the external manifestation. So, for example, blue eyes is the phenotype. And the alleles that make up that trait is called the genotype. So, in the blue eye scenario, you would have a recessive blue eye color gene, for example. But the actual allele combination is called genotype. Okay, the other thing he proposed was that pairs of inherited factors, which we now know are genes, are separated during gamete formation. And this is called the law of segregation. We'll look at this in this lecture, and we'll also look at the other proposition that inherited factors are inherited independently, which is the law of independent assortment. So let's first start with the law of segregation. And I just want to point out here, I'll restate it. It says pairs of inherited factors are separated during gamete formation. Those pairs of inherited factors are the two alleles, right? So you have each gene has two copies. That pair, those two alleles, are separated during gamete formation. So they're separated during meiosis. So let's look at how the law of segregation is explained by meiosis. So here you have gamete formation in two parents. Say parent number one up top is the mother and parent number two is the father. So you'll notice that the mother is heterozygous. She has two different alleles for this gene G. She has uppercase G and lowercase g. Remember, we have two alleles because we get one chromosome from our father and one chromosome from our mother. Now, parent number two on the bottom, the father, he's homozygous dominant, which means that he has two uppercase G alleles, right? And again, he got one uppercase G from his father and one uppercase G from his mother. Now, if you go back up top to parent number one, you can see that at the end of meiosis, the uppercase G and the lowercase g are separated in the gametes. You don't find uppercase G and lowercase g both present in a gamete. If you do, there was some mistake, but normally you will never find that. And this is because of anaphase in meiosis. Anaphase is where the homologous chromosomes are separated, right? Or, if you will, segregated. So the law of segregation is simply talking about how these two alleles are going to be separated or segregated during meiosis. And that results in each allele of the G gene being packed into separate gametes. So this happens in the mother, it happens in the father in the bottom, and these gametes are going to combine at random, right? Random fertilization, and produce two possible combinations of zygotes. So now let's answer a question that is relevant to this. Why doesn't the Punnett square below include a female gamete with genotype RR? Now, I'm not going to ask you to actually make Punnett squares on the test, but I do want you to understand them, and also I would like for you to at least practice them a little bit because they do come in handy. So you'll see up top you have a wrinkled green seed, and so the wrinkled phenotype, which is the outward trait, comes from this recessive R allele. And so this is homozygous recessive for this lowercase r. And it's green, which is the phenotype for the lowercase y allele. So it's homozygous recessive for this allele. So you have RRYY. Now in this plant, the female gametes are going to be lowercase r, lowercase y, lowercase r, lowercase y. Why? Because these alleles segregate, right? They separate during meiosis. Our R separates, and so one R goes into one gamete and the other R goes into the other. Same thing with Y. Supposing Y is on a different chromosome, right? Same thing happens in the male gametes. So in the male, you have yellow smooth seeds, or yellow round seeds. So it's round, so it's going to be, and not going to be, but it happens to be homozygous dominant for this round allele. So uppercase R, and it's going to be homozygous dominant for the yellow allele, uppercase Y. And these also separate. So one uppercase R goes to one gamete, the other one goes to the other. One uppercase Y goes to one gamete, and the other one goes to the other. So now you have the female gametes, the pink eggs up top, and the male gametes, the sperm, on the left. And you can do a Punnett square to figure out the different combinations, the possible combinations of genotypes in the offspring. Now the question is asking, why doesn't the Punnett square below include a female gamete with genotype RR? And now you should understand, or it should be clear to you, that these alleles segregate during meiosis, right? So the answer is because of the law of segregation, and C would be the right answer. The R alleles separate during meiosis. So one R allele ends up in one gamete and the other R allele ends up in the other gamete. That's law of segregation. Now let's look at the law of independent assortment, and this can also be explained by meiosis. So what the law of independent assortment says is that genes on different chromosomes are inherited independently. So in the diagram here, we have a plant, and we're looking at two genes in this plant, the R gene and the G gene. And you'll notice that it is heterozygous for both genes, which means that it has two different alleles, right? It has uppercase R and lowercase r. So it's heterozygous for the R gene, and it has uppercase G, lowercase g, heterozygous for the G gene. So if you look at a diploid cell that's about to undergo meiosis, where you have already replicated the homologous chromosomes, you can see that you have the blue chromosomes are from dad, red chromosomes are from mom. So you have chromosome that has the R gene and chromosomes that has the G gene. Now, one possible alternative of independent assortment is that you line up the chromosomes from dad up top, right? And hopefully this is familiar to you, as we've already covered this in class. And you put the chromosomes from mom on the bottom, the red ones on the bottom. And then throughout in meiosis 1, all the chromosomes up top go to the cell on the top, and all the chromosomes on the bottom go to the cell on the bottom. So then you do another round of division, meiosis 2, and you end up with gametes, with haploid gametes. And you can see that up top, your haploid gametes are uppercase R, uppercase G, uppercase R, uppercase G, and they're both from dad. And on the bottom, you have lowercase r, lowercase g in both cases, and these are both mom chromosomes, right? Chromosomes from the mother of this plant. Another alternative, however, and remember, I forgot to mention, this is at metaphase, right? This is when the chromosomes are lining up in the center of the cell. So another alternative is these chromosomes will have different arrangement. So now instead of just having chromosomes from dad up top, you have a chromosome from dad up top, and then say that's chromosome 1, and then chromosome 2 from mom up top, and the opposite on the bottom. So now when you go through meiosis 1, you end up with a mix, right? Chromosome 1 will be from dad and chromosome 2 from mom, and vice versa on the bottom. And then you undergo the second round of cell division. And now you'll notice that you've mixed things up a bit. So in this alternative, you have each gamete has one chromosome from dad and one from mom. So starting from the first one there, you have chromosome 1 from dad with uppercase R and chromosome 2 from mom with lowercase g. If you go right below it, you have chromosome 1 from mom, lowercase r, and chromosome 2 from dad, uppercase G. So independent assortment just depends on how these chromosomes are arranged at metaphase, and that will create different combinations of these chromosomes in the gametes. So that's why, and it's crazy because Mendel didn't even know about meiosis, right? This stuff didn't come until maybe a century after he did his stuff. But what he saw was that the traits were inherited independently. So the traits that were determined by the R gene and the traits that were determined by the G gene, they could be inherited independently of each other. And we now know that that is because of independent assortment. So let's look at an example in the Punnett square. And again, I'm not going to expect you guys to do something like this on the test, but it helps illustrate independent assortment. So we're looking now at dihybrid crosses. These are crosses that track the inheritance of two genes at once, such as the R gene and the G gene that we've been looking at. So two genes on two different chromosomes can be combined into a dihybrid Punnett square. So in the first look, if we're looking at the first Punnett square on the left, you can see that the female gametes are from a plant that is homozygous recessive for R and for G, and the male gametes come from a plant that is homozygous dominant for R and for G, right? And you cross those plants together, and these are the yellow in the Punnett square are the possible combination of genotypes for the offspring. And you can tell that the phenotype is going to be, for all of them, is going to be round and yellow. And that's because you have the dominant alleles there. Uppercase R is dominant for the round shape of the seed, and uppercase G is dominant for the yellow color of the seed. So all of your seeds are going to be round and yellow, but they're all going to be heterozygous, right? So now if you take seeds from this plant, seeds and sperm, even though plants don't have sperm, pollen, I guess would be a better way to say it, more accurate. And you cross those together, you can see that you now are generating a lot of diversity, right? You're getting a lot of different, or you can make a lot of different possible offspring. So if you look again at the Punnett square on the left, the F1 generation, the parents can only make two possible gametes, right? Because they're homozygous for the R gene and for the G gene. So for example, if you look at the female gametes there, you can only make lowercase r, lowercase g gametes. Those are the only two combinations you can do. But now if you look at the Punnett square on the right, the F2 generation, and you look at the female gametes, now the uppercase R can segregate with uppercase G or lowercase g, right? Those first two gametes up top. And that's independent assortment. So uppercase R does not have to segregate with uppercase G. It can do that with uppercase G or lowercase g. It just depends on how those chromosomes are aligned or arranged during metaphase. Okay, and that's really the point of this slide is the gametes. That's it. I don't want you guys to get too focused on the actual Punnett square, but just realize that the gametes here are illustrating independent assortment. So same thing with, if we just continue with the female gametes and we look at the last two gametes possibilities that you can make, the lowercase r does not have to segregate or assort with the lowercase g. It can also segregate with uppercase G. And this is because they're on two different chromosomes that can arrange in different arrangements during meiosis. Okay. All right, so now let's look at how diseases are inherited, right? This should be interesting because we all probably have a family member that has a disease, and if it's a genetic disease, then you can actually know whether that family member has passed on this disease to, or if there's a possibility that they have passed on this disease to her offspring or his offspring. So most diseases, such as cystic fibrosis in our example below, arise from recessive mutations. Usually recessive alleles are defective alleles. So if you have two defective alleles, then that will render whatever gene we're talking about defective. So recessive mutations require 2 copies of the mutated allele in order to affect the individual. In recessive diseases, the mutated allele is recessive, so it's written in lowercase, and the normal allele is dominant, so it's written in uppercase. So what this means is that even if you just have one normal allele, that person is going to be fine because that normal allele is going to be dominant. So that is what's going to come through in the phenotype, right? So let's continue real quick and look at an example with cystic fibrosis. And this is a disease caused by a recessive mutation in the CFTR gene. Now let's suppose a mother and a father are both carriers of the mutated allele. And they both have one normal allele of the CFTR gene, uppercase F, and one recessive mutated allele of the CFTR gene, lowercase f. So now both of the parents are going to be carriers of cystic fibrosis because they each have one copy of the mutated CFTR allele, right? So they're heterozygous, right? So let's look at the example below. This is from your textbook, and in this figure you have a female that's a carrier. She's heterozygous for the cystic fibrosis gene, and she's going to be artificially inseminated with sperm from a donor that's also heterozygous for the cystic fibrosis allele. And we want to know what is the likelihood or the percentage that their offspring are going to have the disease or are going to be carriers of the disease. So we do a Punnett square for this. Up top we have the possible gametes from the female, and on the left we have the possible gametes from the male. Now the law of segregation states that each allele is going to segregate independently into separate gametes. So up top from the female you have one gamete with the normal allele and one gamete with the mutated allele, and same thing with the father. And now if you combine them using the Punnett square, you'll see that you have 4 different possibilities. The first square top left is homozygous dominant, uppercase F, and that offspring is going to be fine. They have 2 normal copies of the gene, so they're going to be great. Then the upper right and the lower left squares, the light blue shaded ones, are heterozygous, right? They have uppercase F, a normal copy of the allele of the gene, and they have lowercase f, a mutated copy of the gene. So these are going to be carriers. They don't have the disease, but they do carry the mutated allele. And then on the bottom right, you're going to have a homozygous combination of the mutated allele. And this is when the recessive disease comes through. So these offspring are going to have the cystic fibrosis disease, and they're going to be able to pass it on to their children. So now if you look at that percentage-wise, now if you look at the right, 25% it's a 25% chance that this woman's child is not going to have cystic fibrosis, that this child is going to have two normal copies of the cystic fibrosis gene. There's 50% chance that the child is going to be a carrier. So the child won't have the disease, won't be affected by the disease, but he or she will carry the mutated allele and can pass it on to their offspring. And then there's a 25% chance that the child will have cystic fibrosis and will be a carrier. So again, if I ask a question on the exam, what is the chance that these two parents' child will have cystic fibrosis? Now you know you can use a Punnett square and figure out that there's a quarter of a chance that they'll acquire the disease, half a chance, 50% of chance that they'll become an unaffected carrier, and 25% of the chance of not having the disease at all, or the diseased allele at all. Now, dominant diseases are different, and these very few diseases arise from dominant mutations. So, dominant mutations only require one copy of the mutated allele in order to affect the individual. So, they're very dangerous, very prevalent diseases because you just need one copy and you have the disease. In dominant diseases, the mutated allele is dominant, so it is written in uppercase, and the normal allele is recessive, so it's written in lowercase. So, a common example is Huntington's disease, which is caused by a dominant mutation in the Huntington gene. So let's suppose that the mother carries two normal Huntington genes, and the normal gene is going to be lowercase, so she's homozygous for lowercase h. And the father carries one normal Huntington gene and one mutated Huntington gene. So he's heterozygous. He has lowercase h and uppercase h. So what is the chance that their child will have Huntington's? Again, you can use a Punnett square below. You can do a Punnett square and show and figure out what frequency or what will be the chance that their child is going to have the disease. So you align the possible gametes of the mother up top and the possible gametes of the father on the side. And the law of segregation states that the alleles in the mother, who is homozygous for the normal allele, those alleles are gonna separate or segregate into different gametes. So you have one gamete with one of the normal alleles and the other gamete with the other normal allele. And in the father, who's heterozygous, you have one gamete with the diseased allele, the uppercase h, and one gamete with the normal allele, the lowercase h. So you do your Punnett square and you combine, you figure out the possible combinations of gametes, and by that you can determine that there's a 50% chance that their child will acquire the disease, because 2 out of 4 of the possible combinations of gametes have the diseased allele, the uppercase H, and 2 out of 4 have the normal allele, the lowercase h, are homozygous for the lowercase h. So there's 50% chance of not having the diseased allele at all. So again, this is how you could use a Punnett square to figure out the inheritance of a dominant disease. So let's do one last question. And this is same disease model we looked at, cystic fibrosis, but different parents. So we know that it's caused by a recessive allele. And if a healthy carrier, so a heterozygous parent, and an affected individual, which, if it's a recessive disease, the affected individual has to be homozygous for the mutated allele, what is the chance that the child will be affected? So here, what I would want you to do is to draw your Punnett square. So up top, you could say the mother is the carrier, so she has one gamete that has the normal copy of the gene, say uppercase F, and another gamete that has the diseased or the mutated version of the gene, which is lowercase f. And then on the left, you have the father's gametes, which is the affected individual. And so both of his gametes will have the recessive mutated allele. And if you put those together, you will discover that 50% of the time, or there's a 50% likelihood that their child will have cystic fibrosis. All right, now let's look at two more interesting things about inheritance. One is twins. Let's look at how twins are created. So fraternal twins, and these are twins that are not identical, they arise from 2 eggs being fertilized by 2 different sperm. So here in the figure, you see you have 2 eggs, 2 sperm. They get fertilized and they become zygotes, which then develop into embryos and then into a fetus. And basically, this is as if you and your sibling just happen to be in the womb at the same time, right? It's the same thing. They're just occupying the same space, but they're totally different eggs, totally different sperm. So these siblings, these fraternal twins, are going to be 50% identical, which is the same as you and your sibling. Again, each gamete has different genetic material, and therefore each zygote will be genetically different. Now, identical twins arise from the same zygote. So here you only have one egg and one sperm that fertilize to form a zygote. That zygote develops into an embryo, but that embryo, just through its division process, will divide into two embryos. It splits, and those continue the human development process and end up becoming two fetuses and two babies. So these twins are going to be 100% identical, right? They came from the same egg and sperm, so they're going to have 100% of the same genetic makeup, which is why identical twins are identical, right? So now you know how that happens. Okay, so nature and nurture. This is basically, and this is the last slide, and we're just going to ask, do genes absolutely determine who you are? And hopefully, or not hopefully, but maybe you've heard nature and nurture before, nature versus nurture. In this case, nature stands for genes and nurture stands for your environment. So the question here is, do your genes make you who you are, or does your environment make you who you are? And this specifically is asked about things that are very complex, like your IQ or your height or your personality, different behaviors, and things like that. So the most compelling studies are genetic studies using twins separated at birth. And those studies show us that who we are is not only determined by our genes, and that our environment has a role to play. So I suggest watch the video below, because that video, the YouTube link below, takes you to the video, and it talks about these studies looking at twins separated at birth. So these twins separated at birth, they're 100% identical, but they've grown up in different places. And then when they look at them and they look at, for example, their IQ, they're not 100% correlated. So they don't have 100% the same IQ, right? They can vary. So that tells you that it's not, their IQ isn't 100% dependent on their genes. It can also be due to their upbringing, right? And also to the things their lifestyle, what they've eaten, any drugs they've consumed, etc. So we know that the environment has a role to play. However, the floor and the ceiling of certain traits are determined by our genetics. So for example, height. That is how tall you can be is determined by your genetics. The ceiling of how high you can be depends on your genes. And so does the floor, right? How short someone may end up being, there's a certain limitation to that is coded by your genes. So for all of us, the environment can play with where we fall between the shortest our genes allow us to be and the tallest our genes allow us to be, if that makes sense. And again, watch the video below, it's really interesting. So now let's look at the figure on the right real quick. So here there's trying to look at different ways to measure whether someone's IQ is determined by their genes, right? And on the left, you have the method of measurement, and on the right, you have the result. So the first one is the first method of measurement is a correlation between parents' IQ and children's IQ in a population. So for example, two parents have really high IQs, and then you measure the IQ of their children, and you see that there's a certain percentage of their children that have inherited their IQ, right? And so in this study, using this method, they find that 40% of your IQ is inheritable or determined by your genes. The next study, the next method is much better, and this is where you compare identical twins to non-identical twins. And when they do this, they find that when they compare the IQ of identical twins to the IQ of the non-identical twins, there's 50% of the IQ is determined by your genes. But this isn't ideal, because it could just be that the similarity you're looking at there isn't determined by the genes, but simply by the fact that both sets of twins, identical and non-identical, grew up in same environments, right? So it could be that the environment has a role to play here. Now, the best method is when you compare identical twins separated at birth versus non-identical twins separated at birth. And this is great because these twins were raised in different environments. So the identical twins, they have 100% the same genetic content, but they're raised in different environments. The non-identical twins have 50% the genetic content, so they're not identical, right? And they were raised in different environments. So when you compare the two, here you actually see that 70% of our IQ is inheritable, is due to our genes. So 70%, you can look up this study, but don't go up to your parents and tell them, "Hey, gee, thanks, Dad, or Mom, because you gave me 70% of your genes that determine my IQ." Definitely don't do that. But this study is suggesting that that is the case. So again, watch the video and it'll make a lot more sense how they do this. Really interesting stuff. The bullet point take-home message here is that genes do not absolutely determine who you are. The environment does have a role to play. Alrighty. Well, hope you guys enjoyed the lecture, and don't forget to send me your top 2 questions from the test review so that I can compile a little review video so that it can help you study and answer any things that were not clear through the lectures. All right, good luck, bless you. Pray that you would go in peace and in rest. Don't freak out about the exam. Just study hard, do your best, and you'll do well. You will. I was gonna say do your best and God will do the rest, but that's a little cliche, and I'm not sure that's totally theologically, biblically accurate. But I know you guys will do great if you apply yourselves. So see you guys on Tuesday. Bye.